Hello scholars, today we're going to start our journey learning about the physiology tests. These tests can be used to identify unknown bacteria in a patient or environmental sample by matching the unknown test results to the results of bacteria that we actually know. Starting today, we will be looking at these physiology tests, which are all listed here. But in this particular lecture, we're going to be looking at PR fermentation and MRVP alone. Let's start with PR fermentation. This is a differential medium that contains a base broth and a carbohydrate. The carbohydrates are different. In our lab, we have lactose, glucose, sucrose, and mannitol. These are all sugars, and a single carbohydrate is added to it. In the end, you end up with a broth that you would call PR lactose or PR glucose, PR sucrose, or PR mannitol. In lab, when we first started learning about different ways to be able to identify bacteria, we depended a lot on their physical characteristics. Did it have a capsule? What gram reaction did it display? Are there spores? But in front of you, I have a picture of E. coli and E. cloacae, and they are both gram-negative rods. As you can see, doing a gram stain on this does not help us narrow down which organism we have because they look the same. Using physiology tests, these two organisms more than likely will contain different enzymes that allow them to be able to utilize certain things in a medium or not. These organisms may or may not contain certain enzymes that would allow them to utilize what we have placed in the media. The PR test, phenol red, checks for the organism's ability to be able to ferment a particular carbohydrate. And even though one organism can ferment one type of carbohydrate, it may not possess the enzymes to be able to ferment another. With the PR test, we're looking for end products of fermentation, which can either be an acid, a gas, or alkaline. Now let us look at the ingredients of the phenol red fermentation broth. First, we have that single carbohydrate that we mentioned before. There is the base broth, and it is a very versatile medium. And then we add one particular sugar to it to see if the organism can use that sugar. So that carbohydrate is going to be different depending on what we're checking for. Next, we have peptone. The peptone provides a source of amino acids. This is good because then this is an alternative source for energy. What if the organism does not have an enzyme that could help metabolize the carbohydrates? Instead, they will be able to use the peptone. So we're giving them a molecule to utilize just in case they do not use the carbohydrates. Next, there is a Durham tube that is inverted inside of the medium. This is because it would allow us to check for gas production. If gas is produced, a bubble would form inside of the tube. And then we will know that the organism was able to ferment sugar and produce gas. Lastly, there is phenol red added to the medium. This is the pH indicator. Phenol red is yellow if the pH is below 6.8. In other words, if the pH gets acidic, then the medium would turn yellow. Naturally, the phenol red is red. If the organism metabolizes carbohydrates, then it will create acid end products, which will turn the indicator from red to yellow. However, if the organism does not metabolize carbohydrate and instead utilizes the amino acid, the deamination of the amino acids will cause the end product to be alkaline because it will make ammonia. The ammonia is going to raise the pH and it turns the broth pink if no acid is produced. Let us recap what I mentioned on the last slide. If an acid is produced, then the broth turns yellow. If it has alkaline end products, then the broth will turn pink. If the broth is red, this means that the organism did not metabolize the sugar. So we will just consider this no reaction. And if there is a bubble in the Durham tube, we say that it produced gas. With those results in mind, let us analyze the 5 tubes that are before us. Looking at tube A, you can see that it has a yellow color. In addition, there is a bubble inside of the Durham tube. This means that because it's yellow, it fermented the sugar and turned the medium to more of an acidic pH, which causes it to turn yellow. And that bubble indicates gas production. Therefore, A is the formation of acid and gas, and we use the designation Ag. In B, the medium has turned yellow, but there is no bubble inside of the Durham tube, which means that there is only acid production here. We would designate that with an A and then a dash. That dash is negative for gas. C is an uninoculated control, and this is used for comparison because oftentimes it's very difficult to be able to distinguish between the reds that can result in this test. When you look at D, you would see that it has a pinkish color compared to that of the uninoculated control. Therefore, the medium has become more alkaline, and what causes that would be the degradation of the peptone. Therefore, there are alkaline end products, and we provide the designation K. Lastly, if you look at E, it's very close to the uninoculated control. There is no acid and no gas production, so the designation here is negative, negative. The main information that we are deriving from this test is whether it ferments the sugar that's been provided for it and if it produces gas. Now, one thing that I want you to keep in mind is that these organisms can utilize the sugar that's been provided for it only if it has the enzyme. A big mistake that students often make is to think that if organism X with a PR glucose gives this type of designation, so it forms acid and it produces gases, then they think that this is the test result that it should get for every single one of the different PR sugars. But that's not true. This organism for glucose may give a result of AG, but the very same organism, if grown in PR sucrose, can very well give this designation. Nothing is odd about that. All this is saying is that organism X can ferment glucose with acid and gas products because it has the enzyme, but it may not have the enzyme to be able to degrade sucrose. Therefore, we will consider it having no reaction, so negative for both acid and gas. It all has to do with which enzymes the organism has. Our next test on the list is the MRVP. This is methyl red and Voges per scour. MRVP is a combination broth, meaning that it is used for two different tests. It's used for the MR, which is methyl red, and the VP, which is the Voges per scour test. The MR test is detecting if the organism can perform mixed acid fermentation, and the VP detects whether or not the organism ferments glucose and produces acetone and 2,3-butanediol. Let's start talking about the MR test and the theory behind that one. Again, the question is, can the organism perform a mixed acid fermentation? What happens is this media actually has a buffer, and if the organism is able to perform mixed acid fermentation, it's going to overcome that buffer that prevents pH changes, and it would lower the pH. This figure is showing the chemistry behind the mixed acid fermentation. E. coli is a representative positive organism when it comes to the methyl red test. And if you were to need a control when you're running the methyl red test, if you needed a positive control, E. coli would be your best bet. Its mixed acid fermentation produces lactate, ethanol, acetate, and formate, all from pyruvate. Remember, pyruvate is the ending product in glycolysis. And from phosphoenol pyruvate, succinate is made. But the acids that are produced by MR positive organisms are very stable, whereas acids produced by other organisms can be converted to more neutral products. Even if a methyl red negative organism is able to produce acid, the acids that they make are not as stable as the MR positive organisms like E. coli. And their acids would be converted to more neutral products, and you will not get a change in this medium. The MR positive organisms lower the broth's pH permanently. Now let's look at the chemistry of the VP test. Remember, this test is trying to determine if the organism can ferment glucose and produce acetone and 2,3-butanediol. VP positive organisms can perform fermentation and get different end products such as ethanol, lactate, formate, and 2,3-butanediol. The pH drops due to accumulation of acid end products, and this is a signal to switch to 2,3-butanediol fermentation. The dropping of the pH signals the organism to switch to 2,3-butanediol fermentation, and then that raises the intracellular pH. Now, if you look at 2,3-butanediol, even though this is what we're looking for, We're not going to test for it. Right before 2,3-butane-diol, the last intermediate is acetoin. We can test for acetoin. So what we do is we will eventually add something called Barrett's reagent A and Barrett's reagent B. When those are added, they react with the acetoin and it oxidizes it to diacetyl. The diacetyl is going to react with the guanidine, which is already inside of the media, and it would make a red color. So a red color would be positive in this situation. Let's quickly go over the ingredients. We have peptone, which is a protein source with its nitrogen, and of course glucose, because we need some sort of fermentable carbohydrate in there, and the cell loves glucose. And then we also put into this a buffer, potassium phosphate, and this is going to resist the pH change in the medium. The positive organism would literally have to make enough acid to overcome that buffer for it to turn yellow. Now, the MR and the VP are tested for in two different ways. So this is a procedural diagram to help us understand how to do this in the lab. Initially, the MRVP media will be inoculated with pure culture and allowed to incubate for 48 hours. Next, we take 1 ml of the grown-up culture and place it into two different tubes. One is going to be for the MR test, the other is going to be used for the VP. With the MR test, you add 3 drops of methyl red and the color change will be immediate. If the pH is 4.4 and below, then you will get red, which is positive. If the pH is 6.2 and above, then the media will be yellow. Now, between these two shades, you do get an orange color oftentimes. For the MR, the only short positive result would be red. The yellow and the orange are both considered negative. Orange is inconclusive, so we designate it as negative. Now for the VP test. With the VP test, we use two different reagents, Barrett's reagent A and Barrett's reagent B. We'll add 15 drops of reagent A to the culture and we mix it, and then we add about 5 drops of reagent B. Now this test does not change immediately for all organisms. So if you do not get an immediate positive result, then you will monitor this for about 60 minutes, 1 hour. And you would read the result every 10 minutes. Also, gently agitating the tube helps with this. So if you add reagents A and B and it is red, then it is considered positive. If you add reagents A and B and it remains yellow for the first 10 minutes, you still cannot say it's negative. You need to let it go in 10-minute increments. Once it gets to 60 minutes, now you know that you've left this to react long enough. And if it is yellow, it is considered VP negative. If it is red anytime during that 1 hour, it is considered positive. These are what the results look like in the tube. For the MR, once the methyl red indicator dye is added, a red color indicates positive because this means that there are acid end products and the pH is at 4.4 or below. If it is yellow, it is negative, and orange is inconclusive, which we will still consider negative. So if you were in the lab and you got an orange result, then you simply will not use this MR as one of your defining tests to help lead you to your organism. Use something else. For the VP, once the reagents A and B are added, if it turns red during any time in the first hour, it is considered positive. But if it remains sort of a copper color, then we will consider that negative. So that's it for these two physiology tests. If you have any questions, feel free to reach out to me. And in the meantime, happy studying.